All right, so today we're going to be talking about the DNA structure notes. Please note that this is just our academic stuff. This is not honors. That video will be up separately from this. It follows the same basic information, but it is a little bit more complicated than just this. So before we can talk about DNA structure specifically, we have to talk about the 4 biologically important macromolecules. Macromolecules are things that are super important molecules. That's why they're called macromolecules. Macro is going to stand for either very large or very important. So our 4 major macromolecules are carbohydrates, lipids, proteins, and nucleic acids. Now these are the molecule names. A lot of times you'll also hear these as the polymer names. Some of them do have slightly different polymer names, but for the most part they're going to be carbohydrates, lipids, proteins, and nucleic acids. The way that you can think about the difference between polymers, these, and monomers over here, are that they're just like Legos. So if you have just one Lego, it's going to be a monomer. When you have a bunch of Legos put together, it's going to be a macromolecule. So the monomer, the Lego, the single Lego of a carbohydrate is a monosaccharide. For a lipid, it is a fatty acid. For a protein, it is an amino acid. And for the nucleic acids, which are what we're going to be focusing on today, it's the nucleotide. Now, we will be coming back here to proteins and amino acids later on, but for this section, it's just proteins, just amino acids. So inside each and every one of those different inside each one of those monomers, you have 3 different things. So the first is going to be a 5-carbon sugar. In DNA, we use the sugar deoxyribose. In RNA, we use ribose. There's also a phosphate group. For this case, the phosphate group is arguably the least important. It doesn't have that much to do with the general structure. It's just kind of there. You need to know about it, but it doesn't do anything special or different. On the other hand, the nitrogenous base, these are extremely important. The 4 nitrogenous bases are adenine, thymine, guanine, and cytosine. Make sure you can say all of those because you're going to be repeating them out loud in class a lot. So these are only the DNA bases. When we start talking about RNA a little bit later on, that does become different, but for right now, DNA bases, adenine, thymine, guanine, and cytosine. When you look at this, we can see that this is a full this is a full nucleotide because it has a phosphate, a sugar, and a nitrogenous base. A lot of the times we'll say things like, oh, it's a thymine nucleotide, because you'll just see the name for the whole nucleotide based on the name of the base. So when we want to put these together, we have to look at our structure here. So a lot of times you'll hear DNA be called the spun ladder or the twisted ladder. The full name for it is a double helix. So it's double because of the 2 different sides here, and then it's a helix because of the spiral pattern that it makes. So our backbone here, the sides of the ladders, are made up of what's called a sugar phosphate backbone. Now the sugar phosphate backbone is called that because 1, it is entirely composed of the sugar molecules and the phosphate molecules. And it's called a backbone because between each and every one of those sugars and phosphates, you have a covalent bond. Now covalent bonds are some of the strongest bond types in biology. These are not things that are going to be easily broken apart. These are not things that are going to be split apart. The formal name for these structures are the phosphodiester bonds, but for this class, you only need to know that they are called sugar phosphate covalent bonds. You want these to be really strong so that you don't lose any of your nucleotides. You don't want this to be breaking and for this part of the DNA to be flying off in a different direction. You want that to stay exactly where it is in exactly this order. Because it's the order of these bases here, the A's, the T's, the C's, and the G's, that determines what your proteins end up looking like in the long run. All right, so when we are looking between our ladder rungs here, we can see that we have our nitrogenous bases in the middle. The nitrogenous bases in the middle are what make up the rungs of our ladder. Now, unlike the sugar phosphate bonds on the outside here, these bonds in the middle are very weak. You need them to be very weak because you need to be able to rip this DNA in half in order to do anything a little bit later on. So the bonds that hold together these nitrogenous bases are called hydrogen bonds, and hydrogen bonds are just very weak. Sorry, you can hear the printer. Now, if you look up here, you'll notice that these are always bonding in a certain pattern. These have to follow very specific base pair rules. So essentially, these rules are also known as Chargaff's rules, but for this class, you just need to know that they're base pairing rules. So when you look up here, adenine is always going to be bonded to a thymine. Nowhere else are you going to see an adenine bonded to anything else. Thymine is always going to be bonded to an adenine. Guanine is always going to be bonded to a cytosine, and cytosine is always going to need to be bonded to a guanine. And the order of this strand of DNA is really important. This order determines how your proteins ultimately look, and we're going to come back to that concept in just a little bit later. So the very last thing that we need to talk about is the direction of our backbone. So the two strands of DNA are said to be what's called anti-parallel. So when you think of parallel in general, you think of two lines going in the same direction forever and ever and ever without crossing, which is sort of true. These two lines are always going to run opposite of each other without ever touching, but they don't run in the same direction. This top arrow is coming off this direction. This bottom arrow is going off this direction. You can think of this like a highway. You wouldn't want your highways to be perfectly parallel, where they're always going to be going in the same direction. You want them to be going back and forth. That way you're able to come back from wherever your original destination is. What that also means that if one strand starts with a sugar, like our side over here, it has to end with a phosphate. So you're not going to see anything starting and ending with a sugar. It starts with a sugar, it's going to end with a phosphate. By that same token, because they run the opposite direction, if this side starts with a sugar, then this side must start with a phosphate. And if this side starts with phosphate, it has to end with a sugar. The way that I can kind of visualize this is that all of these pentagons are pointing upwards. So they're going up towards the top of the screen, and all of these are pointing downwards. They're pointing down towards the bottom of the screen. So they're running in the opposite direction from one another. So that's pretty much all you need to know for DNA structure. If you have any questions at all, please feel free to let me know. All right, thanks. Bye, guys.